Well, thank you all for coming today. Today I'm going to talk to you about some of my work that is part of my thesis of studying the human microbiome. So some of you may or may not be aware that you're covered in microbes. They're absolutely all over your body. They outnumber your human cells approximately 10 to 1, so they're very, very numerous. And so I've chosen to put this painting by Susie Parker that appeared to USA Today, and it's her interpretation of the human microbiome. And what I thought was so interesting about this painting is how sad the woman looks. I mean, she looks very upset. And perhaps that was many of our gut reactions when we heard we were covered with microbes. It's like, that's really gross. But recent research, I think, has helped us learn that these microorganisms, we've co-evolved with them. They've been with us, from the very beginning, and that they're very important to human health, and they're very important to understand. And so because of that, we'd like to know what influences these microorganisms. And so that's been the focus of my research. And so I study the oral microbiome, so all the microbes that live in your mouth. There's approximately over 700 species. And so this is just a microscopy picture of a scraping from a tongue, and so you can see all the different types of microbes that are present. And so cigarette use has actually been shown to associate with changes in the microbes in your mouth. So people who are habitually using cigarettes look different in their oral microbiome than those who aren't using tobacco. And so with the legalization of marijuana, I became very interested in this question of, will we see a similar shift with those who are using marijuana habitually? Will their microbiome also look different? And how will this compare to tobacco use? So with the generous funding from this grant, I was able to ask that question. So my study population is young adults and adolescents. And so we have a group of people who are non-users, so I call these my goody-two-shoe kids. So these people are using no drugs, they're using no alcohol. And then we have individuals who are only using marijuana and who report only using tobacco, either one or the other, just to help focus the study. And so I asked this question, are different user groups different to each other than the non-users are to one another? Now, I'm not going to go into all the technical details of this specific analysis, but basically we could identify microbes using sequencing. And so here on the y-axis, I have a measurement of diversity. So diversity is just how many different types of microorganisms are present. And I've done this analysis 10 times, and so that's why we see 10 bars for each group. Non-users are in black, marijuana's in blue, and tobacco only is in green. And what you can see is that the people who are using substances are consistently not consistent. They're very, very diverse. They don't look a lot like each other, where those who are not using drugs habitually are much more consistent and stable. And so what we can conclude from this is substance use is associated with changes, and that only using marijuana is showing a similar trend to only using cigarettes. So with that, I'd like to thank all those who helped this project, but especially the Dean's Graduate Student Grant. And I'll take any questions. That's a great question. So in this specific study, it's people only using cigarettes specifically. I do have a collection of people doing chewing tobacco, and I don't have those results yet. This is based on these people were collected over the last 20 years, and so e-cigarettes weren't as popular then, but that's definitely an interesting question for the future to ask. Yeah. Yeah.
Is there a reason to sample young adults in particular?
No. So again, this is just a byproduct of, this is a larger genetic study in which they were collecting adolescents, and so we're now using it to study the microbiome. But good question. Yes?
Responding to resources or stress?
That's an excellent question. So I haven't gotten down to mechanism. Basically, this is just showing change. The hypothesis is that when you're inhaling a lot of the smoke and tar, for example, THC has been shown to be an immunosuppressant, so it's downregulating your immune system. And so the hypothesis is that you have these microbes that have co-evolved with you, the plaque on your teeth, and they're supposed to be there, and that when you're inhaling these substances, it's making it so that they're not able to occupy your tooth niche. And so then you're just getting all these different types of microbes, which include pathogens, and that's why we think we see an increase in poor oral health in people who use these substances. But as far as the exact mechanism, we don't know for yet. But that's my future studies. All right, thank you so much.